Let's look at the first step of cellular respiration, glycolysis. Recall, glycolysis means the splitting of our sugars. Glycolysis is actually divided into two main phases, the energy investment phase and the energy production phase. These two phases are broken down into 10 smaller steps. Each step in cellular respiration, be it glycolysis, citric acid, or the electron transport chain, will show little tiny reactions occurring. This is very important because the smaller the reactions, the more energy that can be conserved and not lost in the form of heat. Glycolysis is no exception. It occurs in the cytosol of the cell, where it is broken down from a large 6-carbon glucose molecule into a smaller 3-carbon sugar molecule. All of the products in glycolysis are organic molecules. When we start off with our energy investment phase, it goes through exactly what it seems like it would. It requires energy to begin. So here we have the 6-carbon glucose molecule. The very first step or chemical reaction in glycolysis is the input of energy. And so we take ATP and we transfer some of the energy into our reaction. As we utilize ATP, glucose becomes now a high energy bond by receiving a phosphate group. It is now called glucose 6-phosphate because it has been phosphorylated. If you recall the molecular structure of glucose, you will recall that the number 6 carbon is outside of its sugar ring. Well, its isomer, fructose, is also a 6-carbon sugar, but it has its number 1 as well as its number 6 carbon on the outside of its sugar ring. Because of this, the next step in glycolysis is to take glucose 6-phosphate with an isomerase enzyme, convert it into fructose 6-phosphate. Because it is now fructose 6-phosphate, the number 6 carbon as well as the number 1 carbon is on the outside of this carbon ring, making it a perfect place for more ATP to come into play. As the second ATP molecule enters into this energy investment phase, we see a second phosphate group attached to the number 1 carbon of fructose. Here we now have fructose 1,6-bisphosphate. Now as you look at this molecule, hopefully you can see that it is one that is going to be able to split apart relatively easily. And so the next step, requiring another enzyme, will actually split this molecule into two 3-carbon molecules. These two 3-carbon molecules, once again, are isomers of each other. One is dihydroxyacetone. The second is glyceraldehyde 3-phosphate. You can tell that dihydroxyacetone is actually a ketone molecule, where glyceraldehyde 3-phosphate has an aldehyde for its double bonded carbonyl. We make both of these from the splitting of glucose in the energy investment phase. However, it is only glyceraldehyde 3-phosphate that can proceed on into the second stage of glycolysis. So as we proceed from this point forward, it is only glyceraldehyde 3 that is moving on. Dihydroxyacetone and glyceraldehyde 3-phosphate are isomers, and so once again a second isomerase enzyme will allow the two to convert back and forth. As we continue on into the second phase of glycolysis, we enter into what's known as the energy payoff phase. This energy payoff phase requires is going to allow us to see the production of our first ATP molecules. We have glyceraldehyde 3 phosphates moving forward. What will happen is an NAD plus will be reduced to NADH plus H plus. But since there are 2 glyceraldehyde phosphates possible, if we look back here, remember we have 2 3-carbon sugars. Only glyceraldehyde 3 will move forward, and so we actually have 2 of them moving forward. So in this energy payoff phase, we're going to reduce 2 NAD pluses to 2 NADH plus 2H plus. But another thing that happens is that 2 inorganic phosphates also come in to play. So what we see is that we now have our phosphate that was on the glyceraldehyde 3 phosphate and a second phosphate come in. That is because there is an open bond on our carbon from the removal of the hydrogens for reducing NADH. Because of this, we see these 2 inorganic phosphates coming into play. We now have 1,3-phosphoglycerate. It is not important that you remember these names, but it is important to remember the reactions that are happening. This 1,3-bisphosphoglycerate is where we're going to produce ATP for the first time. These inorganic phosphates that have come in will be removed back off. Through substrate level phosphorylation, we will see ADP binding to an enzyme and our 1,3-bisphosphoglycerate binding to an enzyme. This phosphate will then be transferred from the carbon to ADP to form this high energy bond of ATP, leaving only 3-phosphoglycerate at the end. Because there are 2 of these, we have 2 reactions and 2 ATPs produced. We then go through some chemical changes, moving the phosphate from the number 1 to the number 2 carbon, and then another enzymatic reaction that allows for a second ADP molecule to come in, and the second substrate level phosphorylation occurs to remove the phosphate once again from 2-phosphoglycerate to form ATP. What is left over is a 3-carbon sugar in the form of pyruvate. Pyruvate is not as high energy bond as 2-phosphoglycerate because the energy has been now trapped in ATP. These 2 ATPs are once again formed through substrate level phosphorylation. So to summarize what we have done, we have had to put 2 ATP molecules in at the beginning of our reaction, and we received 2 ADPs back out. That was our energy investment phase. Through the energy production phase, we have reduced 2 NADHs, we have phosphorylated 4 ATPs, and we have made 2 sugar molecules known as pyruvate. Now, if you recall, NADH has the ability of converting to 3 ATP molecules. So, if we continue through cellular respiration, these 2 NADHs will eventually become 6 more ATPs. Through substrate level phosphorylation, we received 4 ATP molecules, but because we had to put 2 in the beginning, we really truly have only a net gain of 2 ATPs. Here's another way of looking at glycolysis in a full fashion. We start with glucose. Glucose requires 2 ATP molecules and in turn converts it to 2 ADP molecules. We also have 2 NAD pluses and 2 inorganic phosphates that will be reduced to NADH. We then have 4 ADPs going in and 4 ATPs coming back out. So if you take the 2 here and eliminate 2 here, it gives us a net gain of 2 ATPs, 2 NADHs, and 2 pyruvates. Now your textbook goes into all of the chemistry of this. But as you look at this reaction, it should follow a lot easier now. Glucose is a 6-carbon sugar ring. It has the number 6 carbon sticking out on the outside of that ring. The very first chemical reaction that occurs is ATP releasing some energy in the form of that high energy phosphate bond through the help of an enzyme known as hexokinase. We now have glucose 6-phosphate, which will go through an isomeristic change with phosphoglucose isomerase and convert glucose to fructose. Fructose has the number 1 carbon on the outside of its chain, allowing for the next ATP molecule to come in and phosphorylate it. When this happens, there is another enzyme that splits the two in half. As you can see, a very easy line can be drawn right here. We then end up with dihydroxyacetone. Remember, this is the ketone, and that's the second carbon here is the ketone, or glyceraldehyde 3-phosphate, where that phosphate's on the number 3 carbon and the number 1 carbon is our aldehyde. These are two isomers, and an isomerase enzyme brings them back and forth throughout glycolysis. But it is only glyceraldehyde 3-phosphate that can move forward in this pathway. That glyceraldehyde 3-phosphate then is removes some hydrogens and electrons to reduce NADH. Two inorganic phosphates come into play to make 1,3-bisphosphoglycerate. So here's the one that we had. Here's the new phosphate that was just added on. We no longer have our aldehyde. The hydrogen is gone. We now can phosphorylate 2 ATPs. Remember, we've got 2 of these going through. We phosphorylate 2 ATPs. We now have 3-phosphoglycerate. Goes through a enzyme to move the phosphate from number 1 to number 2. And then we can go through a second phosphorylation reaction to turn out with 2 pyruvates, 2 NADHs again, where we can finally have a gain of those electrons. Of those ATPs. Now this is a little bit more complex, but if you can follow the other 2 pathways, this is able to make a lot more sense. I'm not requiring you to understand all of the chemistry involved, but you do need to know the general reactions of what is happening here. Once again, to summarize, we start off with glucose. We must add 2 ATP molecules to the reaction to start it off. That is our energy investment phase. We then are able to make 4 ATPs, reduce 2 NADHs, and have left over 2 pyruvate molecules. Because we had to put 2 in, when we get 4 ATPs back out, we have a net gain of 2 ATPs, 2 NADHs plus 2H plus, and 2 pyruvates. Now if we watch a short little video on what's going on here, we will be able to see this glycolysis in process. Recall that glycolysis occurs in the cytosol. For the initial start of glycolysis, we must have 2 ATP molecules. These 2 ATP molecules are involved in the energy investment phase. So if we watch a simple animation on this, we can see how ATP comes in and phosphorylates our glucose. The first one went on the number 6 carbon. The second one came on the number 1 carbon. We now have fructose 1,6-diphosphate. We invested 2 ATP molecules. Now we are capable of splitting the glucose molecule in half to form dihydroxyacetone and glyceraldehyde 3-phosphate. But since glyceraldehyde 3-phosphate is the only one capable of proceeding forward, the dihydroxyacetone will go through an isomerase enzyme to create glyceraldehyde 3-phosphate as well. Now we can see how these two 3-carbon sugars can actually go into producing or forming 4 ATPs. First, we reduce NADH and attach an inorganic phosphate. Then ADP is phosphorylated to form ATP. These 4 ATPs that are produced are formed through substrate level phosphorylation. We have a net gain of only 2 ATPs because we needed 2 ATPs to start the entire reaction. So if we watch this again, ATP comes in, phosphorylates our sugar. We then split our sugar in half, convert dihydroxyacetone to glyceraldehyde 3-phosphate, and then we see the reduction of 2 NADHs and the phosphorylation of 4 ATPs.